Cool, so welcome back, and we're continuing on with the steps in our scientific methods. So in the previous video, we asked our research questions, so we're interested in Serena amelia. We did a bit of background research and we identified 2 candidate genes that might be involved in causing this phenotype. We formed a hypothesis and an objective or an aim, and now we've got to test that theory with an experiment and create some data of our own. So what we've got to think about is how we're going to achieve our aim here and how we're going to test our hypothesis. So what we need to consider is methodology. So we need to design our experiment, so we need to have some groups, perhaps a control group where no genes are ablated or knocked out, and an experimental group where we do something to these mice. We might get rid of the genes, TSG and BMP7. We then need to prepare our experiment with an animal model, or perhaps we could use a human model. And we also need to take some measurements, so how we're going to measure this. And also you finish off your experiment with some statistical analysis. Now the last 2 points aren't too important for you in second year, but it's good to have them in mind and to consider this when we're designing an experiment. So the things we need to really consider are that our model is relevant and applicable. So we're interested in limb development, more specifically hind limb development or lower limb development. So we can't do this in a model that doesn't have any limbs. That's not very realistic. So we can't do this in perhaps a C. elegans or a worm model. Can we do it in human or animal? In humans or an animal? So there are things we can do within a human model of experiment that we can't do in an animal, and vice versa. Humans can give their consent, animals cannot, so there are tight regulations about which animal model we can use. For example, using a non-human primate to using a Drosophila fruit fly. There are ethical considerations in place. So the big difference between using a Drosophila or perhaps using a non-human primate, so we'd use something like Drosophila if we're interested in perhaps genetics and genes. We'd use a non-human primate perhaps for something that is not very well conserved across species. So if we're interested in perhaps kidney development, the kidneys of a human are not very similar to any other animal model. They do share some similarities, but it's not very well conserved. So these are other considerations you need to have in mind. The other thing is when we're deciding on an animal model, we need to think about the relevance of the animal model to a human if we're interested in a human pathology. We also need to think about gestation. So what's the gestational length? So mice gestate for about 19 days, humans about 9 months. Are these similarities comparable? And it depends on what we're studying. So if we're interested in pregnancy, gestation might be a quite important thing to consider. Lifespan is quite important, so the turnover. Fruit flies you can have for a couple of days. Sheep, on the other hand, might last you a couple of years depending on what you're interested in. The anatomy and physiology of the animal is a big one, so we need to be considering the anatomy and physiology of the animal and whether or not it's relevant to what we're interested in. The genetics, so are the genes conserved in this animal model compared to a human? The availability of the animal, is it going to be easy to acquire this animal? With mice you can order the knockouts online with a click of a button. With sheep it's a bit harder. With non-human primates it's very difficult to obtain these animal models. There's also the cost of housing these animals, feeding the animals, also providing them with adequate veterinary support as well. Their litter size is quite important, so if we're interested in pregnancy, in a mouse, they can have litters of up to 8 mice, and this can significantly reduce some of the embryo sizes because they're not receiving as much blood as the rest of the embryos. So these are all important considerations to have when we're deciding on an animal model. So which model of development should we use to study Serena amelia? So we're interested in a congenital abnormality, which means a prenatal stage of development. So we're interested in the developing embryo before birth. We need a vertebrate model. We're interested in lower limb development. So we need to manipulate some genes here as well. So we can't do this in a human. We can't do this in a non-human primate. We can't alter the genes of a sheep. A good idea might be to knock out these genes or completely get rid of the gene, or perhaps knock them down and reduce its function. Now we can do this in a mouse, and it's kind of the go-to gold standard in research. So a knockout mouse is a great idea, but why would we use one? Well, the genome of a mouse is fully sequenced. It has a similar anatomy and physiology to that of a human. It's not the same, it might not look it, but it is very, very similar. And it's similar enough for us to be able to use it for a limb development model. Their bones develop in the same way that ours do. They develop through intramembranous and endochondral ossification. They have lower limbs or hind limbs, so it's something we can use. They have long bones. They're easy to house and obtain, so mice across Australia are housed in animal housing facilities, and these are quite common to come across. And the people who house these animals are very used to handling these mice, and they're not very difficult to use. They're a vertebrate mammal, like us, so that's always a good thing, and they have a similar skeletal development. And they also develop in utero like we do. They have a reasonable gestation time, so about 19 to 20 days, so it's not going to take us a whole year to complete this experiment, hopefully. 19 days, we should have a turnover of these mice. And they also have highly conserved genes, very similar to us. And they're quite easy to manipulate, so we can surgically manipulate them, we can inject them with some sort of substance, or perhaps we can get rid of some genes. It's very, very easy to use a mouse model. So how do we quantify our intervention? So we can look at the gross morphology of a mouse, meaning we can look at just the mouse itself to see what it looks like. If we're going to knock out these two genes, both BMP7 and TSG, and we're interested in whether or not it's going to perturb lower limb development, we can just look at the lower limbs and see if there's anything wrong with them. We could then look at the tissue itself, so stain the whole embryo or fetus using histology, perhaps using some immunofluorescence or immunohistochemical staining like you've learned in your previous lectures. And then the end goal here is that our analysis should be relevant and contribute to answering our hypothesis. So let's go over our plan here. So we want to generate a double homozygous knockout mouse. So we're going to get rid of BMP7 and TSG in a mouse model. We're going to observe the phenotype of the offspring, so look at the gross morphology of it. We're then going to do a tissue analysis, so perhaps we can stain the whole embryo. We can stain it with Alizine red to stain the bones and Alcium blue to stain some cartilage. We can then analyse our results from this tissue and morphological analysis. So how the researchers in this study went about generating the double knockout mouse was they mated a BMP and TSG mutant, and they crossed it. The resulting fetus was a double heterozygote knockout, so had one copy of BMP7 and one copy of TSG. They then crossed those mice, and the resulting phenotype was the F2 generation, and they got a whole range of different genotypes. So some of them had one copy of BMP7, some of them had no copies of TSG, and the one we were interested in is having absolutely no copies of either BMP7 or TSG present. So that's the end of the second video, and I'll see you in the next video where we'll discuss the results of this study.